students from Del Norte High School. For our biology project, we had to create an ecosystem. This ecosystem included a terrestrial chamber, an aquatic chamber, and a decomposition chamber.
For the past couple weeks, we've been collecting data and observing how biotic and abiotic factors can create an ecosystem and depend on each other.
Parallel to our ecozones, we've been learning and studying what we have been observing from textbooks.
During these past few weeks, we have also been taking high-res pictures and videos of our ecozone and the organisms thriving inside of it.
This is the equipment we use to collect our data.
This is our aquatic chamber we have been working weeks on. To the left is our terrestrial chamber, to the right is the decomposable chamber, and that over there is the aquatic chamber with our fish.
This is the betta fish that we put into our aquatic chamber. If you look, you can see how it moves around and the texture of its body by looking at its scales.
Other than the visible organisms, there are also small components of bacteria in our ecosystem. We found these on the leaf in the decomposition chamber.
These bacteria were found in the moisture on the walls of the terrestrial chamber.
In our aquatic chamber, we had one fish, one plant, 20 centimeters of water, and 5 centimeters of gravel, along with rope inside the water, 3 centimeters deep. In the terrestrial chamber, we had 10 centimeters of soil, a rosemary plant, 3 snails, 3 crickets. We also had 2 spiders. One was about 2 centimeters in width, the other about 4 centimeters in width, with an egg sac. Later, 3 egg sacs were made. There were also 5 mealworms. In the decomposition chamber, there were 10 pieces of fruit, 30 leaves, 10 chunks of potting soil, 15 mealworm, 2 decomposing worms, and 6 roly-polies.
On September 3rd for our aquatic chamber, the volume of the water was 18.5 centimeters. Our conductivity was 109 US, and the pH was 5.5, and the dissolved oxygen was 1.5. Fish looks mobile and plants look healthy. For our terrestrial chamber, the soil temperature was 22.5 degrees Celsius, and the air temperature was 23.3 degrees Celsius. The barometric pressure was 29.28, and the relative humidity was 100%. The carbon dioxide ppm was 39,000 and the oxygen ppm was 137,000. The snails and spiders are living and healthy. For our decomposition chamber, the roly-polies are visible and there's limited decay. And the temperature was 23.8 degrees Celsius.
September 9th, in our aquatic chamber, the volume of water was 17.9 centimeters. Our conductivity was 123 US, our pH was 5.54, and our dissolved oxygen was 0. The fish looked healthy, and there was lots of bacteria growing on them. In our terrestrial chamber, the soil temperature was 22, the air temperature was 22.7, our barometric pressure was 29.37, our humidity was 100%, our CO2 was 390, our O2 was 157,755. The snails, spiders, and plants looked healthy in our light. In our decomposition chamber, our temperature was 23.8. Organic matter seems to be decomposing, and the roly-polies are not visible.
On September 15th, in the aquatic chamber, the volume of the water was 17.4 centimeters. The conductivity was 135 US, pH was 5.86, and the dissolved oxygen was 0. There was lots of bacteria on the plants, and the fish is alive. The plants look healthy. Also, in the terrestrial chamber, soil temperature is 22.1, air temperature is 23.2, barometric temperature is 29.35, relative humidity is 100%, CO2 is 76,000, oxygen is 169,478. Plants are decaying, snails and spiders look alive. In the decomposition chamber, everything is starting to decompose and life is not present. All of the beautiful nature that you have seen in our ecosystem and nature that you see every day in your community is being threatened by human waste that is toxic to all organisms around the biosphere. We can all help by recycling, reducing waste, and minimizing plastic and paper usage.
Reduce, reuse, and recycle!